Hello everyone, welcome back to my channel Giemsa. Today we will understand about Mycoplasma. So what is Mycoplasma? Mycoplasma are the smallest free-living prokaryotes, typically measuring about 0.15 to 0.3 micrometer in diameter. They belong to the class Mollicutes, which literally means soft skin, referring to their lack of a rigid cell wall. Because they do not possess a cell wall, they are naturally resistant to beta-lactam antibiotics. Now, what are the key features of Mycoplasma? These organisms have a sterol-containing plasma membrane, which provides structural stability in place of a cell wall. They are pleomorphic, meaning they can take on various shapes. Mycoplasma also have extremely small genomes, ranging from about 580 to 1350 kb, and they require cholesterol for their growth due to their unique membrane structure. One important term we need to understand: what do you understand by sterols? So, sterols are lipid molecules that help stabilize cell membranes, especially in organisms lacking a cell wall. Now, how do mycoplasma behave in the laboratory? In the lab, mycoplasma grow on PPLO agar supplemented with serum to supply necessary sterols. PPLO means pneumonia-like organism. Their colonies have a characteristic fried egg appearance with a dense center and lighter periphery. They do not gram stain because of the absence of a cell wall, so Giemsa staining is preferred. Their growth is slow, often requiring 1 to 3 weeks for visible colonies. Which mycoplasma species are clinically important? Mycoplasma pneumoniae causes atypical pneumonia and associated with production of cold agglutinins, which are antibodies that agglutinate red blood cells at low temperature. M. hominis and Ureaplasma urealyticum are linked with genitourinary infections or GU infections, including pelvic inflammatory disease or PID and urethritis. M. genitalium is another important cause of non-gonococcal urethritis or NGU and is known for rising antibiotic resistance. Now here another term is very important, that is cold agglutinins. So these are autoantibodies, usually IgM type of antibody, that cause red blood cells to clump in cold temperature. How is mycoplasma diagnosed? PCR testing is the most important, sensitive and a specific method for detecting mycoplasma infections. Serological tests detect the IgM or IgG antibodies may also be used. The cold agglutinin test is positive in roughly half of the M. pneumoniae cases but is not fully reliable. How are mycoplasma infections treated? Macrolides such as azithromycin and erythromycin are the first line treatments. Tetracyclines and fluoroquinolones are effective alternatives. Since mycoplasma lack a cell wall, beta-lactam antibiotics and vancomycin are completely ineffective. Now, what are the key points we need to remember? These bacteria lack a cell wall, making them naturally resistant to beta-lactam drugs. They are smallest free-living bacteria and form characteristic fried egg colonies. In culture, M. pneumoniae is an important cause of atypical pneumonia and is associated with cold agglutinins. Macrolides and tetracyclines remain the treatment of choice. If you like this video, please do like, comment, share and subscribe. Thank you for staying with me.